The next two chapters are microbial genetics and viruses, and I'm lumping these together into 1 week because viruses are just a bit of genetic material, and so I think they fit really nicely in with microbial genetics, but in your textbook these are two separate chapters. So chapter 9, this is going to be mostly bacterial genetics. So I'm afraid I cannot, this chapter is mostly prereq review, mostly. So for those of you who've had your prereqs in a reasonable amount of time before this class, this chapter should be fairly easy, chapter 9. If it's been quite a while since your prereqs, you're going to maybe be scrambling a bit. But I'm afraid that I can't teach two classes at once. I do my best, right? I do my best to do as much prereq material as I can, but there's a reason for the prereqs, and it's because I can't fit two classes in one. So I have to move forward from the prereqs, and I know a lot of people have been feeling like it's a lot of material to be doing, like, the electron transport chain felt huge and stuff like that. But the chapters that have review, I do my best to teach them from scratch, but if you are trying to get this class in without doing prereqs that you had a long time ago, it's just going to be a little bit more work, okay? So we'll review what DNA is, we'll review transcription and translation, we'll review what mutations and mutagens are. Really the only new thing in this chapter is horizontal gene transfer. And then maybe pre and post-transcriptional controls, which has to do with how the DNA code is expressed outwardly. We'll talk again about diversity and evolution, which we talked about in chapter 1, but we'll add on to it now that we're covering genetics. All right, so DNA stands for deoxyribonucleic acid. Deoxyribose is the name of the sugar in DNA, and then nucleic acid, that's what kind of molecule it is. Just like proteins are made out of amino acids, DNA and RNA are made out of nucleic acids. So DNA stores the instructions for building an organism. It's the blueprint for life. Okay, so like you get a new Lego set and it's Lego Star Wars, and it gives you like this instruction book for how to build all the stuff. DNA is the instruction book. Okay, for building an organism. It doesn't matter where the Legos or the building blocks come from, the instructions always build the same thing. One set of instructions will always build the same thing. DNA is passed on from parents to offspring, right? So for clonal species, the DNA is passed precisely, right? I mean, aside from mutations, but the DNA is copied molecule by molecule, nitrogenous base by nitrogenous base, letter by letter, to the offspring. So the offspring are the same as the parents. They're clones of the parents. For those of us who engage in sexual reproduction, we're not clones of our parents, obviously, but we still get our DNA from our parents. So I am a mix of my mother and father's genes, right? All my DNA comes from my mother and my father, but I'm a unique mix of my mother and father's genes. So when I'm making eggs, to make eggs, my father's genes that I carry and my mother's genes that I carry get randomly shuffled and placed into each egg. So each egg is a unique mix of my father's genes and my mother's genes, right? And my children are 100%, their DNA is 100% from their father and me, from nobody else. So we still give our DNA, my children couldn't just be anyone, right? Because they have to choose, their genes have to come from me and their dad. But we don't know what shuffle of genes they're going to get from the two of us. Right? All living organisms contain DNA. Right now you get into that gray area between living and non-living, and that you find some other kinds of instructions involved, some other molecules used for replication. But for living things, cell and up, it's always DNA. All right, DNA is a double helix. Double means two, so you can see that there's two strands. So if you look at these blue balls, these strings of blue balls, there's two of them, and they wind around each other. Helix is just another word. It's a kind of spiral. So it's a double spiral. So there's two strands of DNA that spiral around each other. So that's a double helix shape. DNA is composed of nucleotides. So we call DNA a polymer. Proteins are polymers, DNA polymers, starch, glycogen, those are polymers, those are carbohydrate polymers. So a polymer is a large molecule made out of repeating subunits, and I like to use a pearl necklace as an analogy. So if you had a pearl necklace, the whole necklace would be the polymer, and this repeating subunits are the pearls. So a pearl necklace is just pearl, 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 all strung together. right? So each pearl is a monomer. Mono means one. So each pearl is a monomer, and the monomers are strung together repetitively to make the polymer. Poly means many. Mono means one, poly means many. So a protein is a polymer made out of stringing amino acids together. So the amino acids are pearls for proteins. For starches and glycogen, the monomers would be carbohydrates, or excuse me, would be sugars. So sugars are strung together to make these longer carbohydrates like starch and glycogen. Okay. And then for DNA and RNA, for these nucleic acids, nucleotides are the pearls that are strung together. So here's one nucleotide, and these get strung together in these long, long, long chains for 6 feet long in human cells to make DNA. So inside each of your cells, you have a 6-foot long molecule of DNA, and it's just these repeating over and over and over again, strung together. Okay, so there's 3 components on each nucleotide. There's a sugar, and this is deoxyribose for the D part of DNA. So deoxyribose sugar, it's a 5-carbon sugar. Then a phosphate group, and that's phosphorus surrounded by oxygens. And then you have a base, a nitrogenous base. Nitrogenous bases, there's 4 kinds: adenine, cytosine, guanine, thymine. Adenine, for A is for adenine, C is for cytosine, G is for guanine, T is for thymine. These are the 4 different nitrogenous bases of the genetic code. So there's 4 quote-unquote letters. They're not really letters, obviously. There's no letters in molecular letters in your cells. These are the abbreviations of the name of the molecule. So there's these 4 molecules, but we can call them they're kind of like letters because the arrangement of them spells out a protein, you know, like something that the DNA is a blueprint for. So they're kind of like words in a way. So the unique arrangement of these 4 nitrogenous bases from gene to gene, that's what determines what's built from the gene. Okay, so this is the code. This part, the nitrogenous bases, make the code. So if you look at this molecule over here, the greens, pinks, blues, oranges, that is the genetic code. So that's the part that, and we've cracked the code. We know what it is. We can sequence it, and then we can figure out what protein it builds because we know what the letters correspond to. Okay, so we've got this code cracked. So we can read DNA and know what it says. We can even genetically modify things and alter and do funky things with the DNA to make it. We're still learning, but we really understand a lot about this genetic code right now. All right, the sugar phosphate groups, that's the backbone. So it's like the binding of the book. It's just structural. It holds the nitrogenous bases together in the right order so that the code can be preserved. So here's the code. That's like the letters or the words. And then here's the sugar phosphate backbone that just holds them all together. So you can see sugar phosphate, sugar phosphate, sugar phosphate, repeating over and over again. So these nucleotides are strung together by the sugars and phosphates, and then the nitrogenous bases stick out in the middle, and they sort of make the rungs of a ladder. So you can sort of think of DNA as like a ladder where it was able to be twisted upon itself to make that helix shape. But the nitrogenous bases stick out towards each other. They form hydrogen bonds with each other. They're the rungs of the ladder. All right, so DNA is directional. It has directionality. So again, I'm going to make this analogy with words and letters. So you know that when you read a book in English, you read left to right. And if you try to flip that book around and look at it in a mirror, and all the words are backwards, it's really much harder to read, right? Because the words don't say the same thing forwards as backwards. If you read the words backwards, they'd make a totally different sound, totally different word, right? Usually a nonsense word. Very rarely a real word. It's usually just nonsense if you try to read it backwards. Well, it's the same thing with DNA. The enzymes that read the genetic code and build something from it, build cell parts from it, they need to know which direction to read. Because one direction makes sense and it will help it, the blueprint, if it's being read the correct direction, it gives proper instructions, the sensible instructions for a cell part. But if you read it backwards, it's just gobbledygook, right? Because the letters aren't the same forwards and backwards. So the enzymes need to know which direction to read. They're directional. So we have a 3' and a 5' end on each strand. So we tell which direction by the sugar phosphate backbone. So in these pictures, it's not shown here, but this tip Any picture I give you on a quiz or an exam will show the oxygen. This peak here, that's an oxygen. So you can count the carbons from the oxygen to know what direction is. These oxygen points one direction. So you have, counting this direction, you have, so oxygen, counting clockwise from the oxygen, you have carbon 1 on this corner, carbon 2, carbon 3, carbon 4, carbon 5, and then the phosphate group. So here's one nucleotide here, right? The phosphate group is pointing up. Then you have the sugar attached to the nitrogenous base, which is going to be an A, G, C, or T, right? Adenine, thymine, guanine, or cytosine. So you can count sugar, carbon 1, just on this sugar, just on this 5-carbon sugar, you count. This is an oxygen at the peak. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Carbon 5 is attached to the phosphate group of the same nucleotide. Carbon 3 is attached to the phosphate group of the previous nucleotide. Okay, so you can notice that these two strands are in opposite directions, right? This is one strand. All the oxygens are pointing up, and the phosphate groups are pointing up, right? This phosphate group is from this nucleus. So this is, if you start here, this is starting at the bottom. The phosphate groups are pointing up. Here, the phosphate groups are pointing down. Right? The phosphate groups are pointing down, the oxygens are pointing down. So enzymes know which way to read based on the directionality of the sugar-phosphate backbone. This one is sugar at the bottom, phosphate at the top. This one is sugar at the top. You know, you can't really, this phosphate is off the slide, but sugar at the top, phosphate at the bottom. Sugar going down to phosphate. Then the next one, sugar down to phosphate. Then the next one, sugar down to phosphate. Okay. And you can also look at the direction of that oxygen at the peak of the sugar and which direction it's moving. So that tells the enzymes which direction to read. So we call these the 3' and the 5' ends. Okay. So again, counting, you know, if you're counting the sugar here, so here's the oxygen, counting the carbons on the sugar. So oxygen's here, carbon 1, carbon 2, carbon 3. Carbon 3 is pointing down. That makes the down the 3' end. Carbon 4, carbon 5, phosphate. Carbon 5 is pointing up. That means this at the top over here is the 5' end. 3' here, 5' here, because they're all oriented the same way, right? It doesn't matter which sugar you count, it's the same. So here's the oxygen, carbon 1, carbon 2, carbon 3. Carbon 3 is down. Carbon 4, carbon 5 is up, right? So the downside on this slide is the 3' end. The upside on this strand is the 5' end. Now, the other end is upside down relative to this, right? So here's the oxygen, and you count clockwise. So carbon 1, carbon 2, carbon 3. Carbon 3 is actually up. Carbon 4, carbon 5 is down. 5' end is down here, 3' end is up. So they're opposite each other. So you can remember that the 5th carbon is attached to the phosphate group. So phosphate group points to the 5' end. The sugar is on the side of the 3' end. Okay. So we can see that here too. Sugar, phosphate, sugar, phosphate, sugar, phosphate. So down here, this is the 3' end. Up on the top of the strand, that's the 5' end. Enzymes, the enzyme that reads DNA, all the enzymes that read DNA are going to read 3' to 5'. Okay, so they always travel from here to here. And they're going to move backwards along the other strand, right? Because the other strand is upside down relative to, the two strands are upside down relative to each other. So the enzymes are going to travel backwards on the other strand. Okay. All right, so DNA sequences can be represented by the letters of their bases. So we don't tend to draw the whole DNA molecule. We don't care so much about the sugar-phosphate backbone. We care about the A's, T's, G's, and C's because that's where the code is. When we read that, we know what is being encoded by the DNA gene. So we can read this, and if we only need to, so DNA sequencers, you're just sequencing one strand at a time. So you have to unravel the strands before you can sequence them in our new finagled equipment these days that read DNA sequences. You have to unravel them. You only read one strand at a time. But if you know one strand, you can figure out the code of the other because of what we call complementary base pairing. Okay, so let me read this first before I move on. Different patterns of nucleotides code for different proteins, the way different patterns of letters code for different words. Right? So you might think, okay, how can all the diversity of life be encoded in just 4 symbols, 4 molecules, 4 letters? You know, we have a 26-letter alphabet that makes our entire language. How can you have something so complex with only 4 letters? Well, think about a computer. A computer runs off of a binary code, just ones and zeros, just 2 symbols. DNA has 4 symbols. That's much more complex. And the words can be, there's no limit on word length, right? So each code for each protein blueprint, it can be as long as, you know, whatever. It can be 100 nucleotides long, it can be thousands of nucleotides long. There's no limit on length. And so the number of combinations of letters you can come up with is, you know, practically, well, there's no such thing as effectively infinite, but you know what I mean. Like there's not really a limit on how, there's essentially, you know, for all practical purposes, no limit on the combinations you could produce. So different patterns of nucleotides code for different proteins, because DNA codes for protein, the way different patterns of letters code for different words. So I already talked in a previous lecture that DNA codes for protein. It doesn't code for anything else in the cell. It only codes for proteins, and proteins build all the rest of the cell. So proteins are the little dynamic robots that keep the cell maintained. They build things, they break them down, they let things in and out of the cell. They are defense and communication molecules. The proteins are little robots with specialized function that keep the cell, they maintain the cell. Okay, they build the cell. DNA codes for proteins, proteins do the rest. Okay, so different patterns of nucleotides code for different proteins, the way different patterns of letters code for different words. And there's complementary base pairing. So like I said, if you know the code for one strand, you can figure out the code for the next through complementary base pairing. So A's always bind with T's, always. A's and T's always bind. T's always bind to A's, G's always bind to C's. So if my code for the strand that I know is ATG, I can infer that the complementary strand that it usually attaches to is TAC, because T's aren't going to bind with G's or C's. They're only going to bind with A's. A's and T's only bind. Only C's and G's bind, right? Okay, so that's called, so these 2 strands are not mirror images of each other, but they're complementary. So if we know the code of one strand, we can figure out the code of the next. And I'll show you, I'll remind you, so here's a little 3s and 5s on the corners here. That's to indicate directionality, because again, it doesn't do you any good to just see a string of DNA letters because you don't know which way the enzyme's reading it. Is it reading it this way? Is it reading it this way? I want you to forget left and right. I know that you, as an English or Spanish-speaking human, you are used to left-right, right? So some of you might also be proficient in other languages that don't use the left to right direction for reading, and maybe that will help you. You need to get out of that English-speaking human mindset. DNA has nothing to do with your language. It predated your language by billions of years. So there's no left or right. There's just 3' to 5'. So here's 2 strands, and they each are going to have different codes at different parts of the DNA strand. The enzyme's going to read the bottom strand right to left, and the top strand left to right. Because it doesn't matter left to right, it matters 3' to 5'. This strand is read in this direction because this direction is 3' to 5'. Starting here is the 3' end, and it travels to the 5' end. The enzymes that read the top strand are going to move in the opposite direction because the top strand is what we call anti-parallel. Anti-parallel. So that's another vocabulary word. It's in your textbook. Anti-parallel. Parallel means that the 2 strands are equidistant apart through their entire length, right? So if we went to one of these, to this molecule here, to this image here. So if you were to measure the distance between the 2 sugar phosphate backbones, they're always going to be the same distance apart through their entire length. It's not like it's wider some places and narrower some places. The 2 backbones are parallel. The 2 strands are parallel. They are equidistant apart through their entire length. But they are anti-parallel because the 2 strands are upside down relative to each other. Okay, the backbones travel in opposite directions, which is what we talked about here. Sugar phosphate backbone facing up here and facing down over here. So here's the 3' to 5' end in this direction, 3' to 5' end in that direction. You need to know where the 3' is and where the 5' is if you're going to know what protein, if you're going to be able to read this and translate it into a protein, you need to know which direction is which. You need to know where the 3' starts and where the 5' ends. All right, so the vocabulary words that we've covered are antiparallel. You need to know what antiparallel means. That refers to the sugar phosphate backbone and its opposite directions of each backbone. The two backbones are parallel, but they run in opposite directions. Okay, so antiparallel, you've learned about complementary base pairing rules, A's to T's, G's to C's, and you've learned double helix. Okay, that's the double spiral shape.